Okay, yeah, so thank you everyone for coming. It's great to see all you here. So, and thank you Tobias for the introduction. What I want to talk about today is a bit of an overview of where we've got to where we are now, and this long-term interest in this protein called Talin, and how I've been following this for many years now, since 2005, and trying to uncover what this is doing inside of our cells. So the title was deliberately broad, but what I want to talk about is how we got to understanding this molecule and how the knowledge of this molecule has led to a deeper understanding of biology, but also the translation into a new type of materials using these special properties of the molecule I'm going to show you to make new classes of materials. So what we're interested in the lab is this process of cell matrix adhesion. All the cells in our body are attached to to each other and to the extracellular matrix. They're not just a big ball of amoeba. We've got highly structured, highly organized tissues. And this cell shown here is demonstrating the formation of these adhesion complexes. In the background, which you can't see in this, is a complex meshwork of proteins called the extracellular matrix. And this guy, this fibroblast cell, is crawling along that surface. And what you see is in green is where this cell is physically engaging and making these adhesive structures. And our research is to understand these green assemblies. And the reason the cell can grab onto the extracellular matrix is because on the surface of a cell is a class of receptors, well, there's other ones, but called the integrins. And the integrins are essentially the hands of the cell. They can physically, when they're activated, grab on to the extracellular matrix, or they can let go by going into this low activation state. And Talin, the protein we're going to talk about today, sits just inside of the cell, and it can pull on these short cytoplasmic tails of the integrin, and in doing so stabilize this active conformation. In the simplest sense, when Talin binds to integrin, it switches the integrin on. But Talin does a whole lot more than just activate and control the activation state of the integrins. It also couples this integrin matrix complexes to the cell's fourth generation machinery and the cytoskeleton. And it does this via direct links to actin and indirect links to actin and microtubules. And the classical view of Talin's role here is that it serves to sense mechanical forces, but the classical view of that is that in response to force, this molecule experiences tension and it recruits a protein called vinculin. And vinculin is shown in green here, and it's also the molecule which is fluorescently tagged here. This is when vinculin binds. And Talin is also perfectly positioned in this to sense forces from both outside of the cell via the matrix and the integrin complexes, but also via the fourth generation machinery. And what's become apparent, certainly with the advent of cheap sequencing, is that there's hundreds of diseases which originate from where these adhesive structures go wrong, and it impacts on pretty much all disease conditions, mutations linked to mess in this signaling complex. What I want to draw your attention to here is this is just four proteins. This is an integrin with a Talin connected to actin with a vinculin, which is cross-linking this together. But onto that simple core of four proteins, we can assemble up to 280 different proteins will come and bind to this. And that gives it this ability to do all sorts of different cellular functions, not just in the cell migration shown here where this cell's crawling along, but here we've got a Drosophila embryo which is just becoming contractile. It's less than about a millimetre long. But in yellow, again, we've got the Talin integrin actin complexes which are holding these myotendinous junctions together. And it makes all of these structures, which are very important during in cancer and in metastasis, these invasive podia structures which can burst through and get to other places in the cell. But what we're very interested in are these two on the right-hand side. This is a cell which is just undergoing mitosis. And if you look just before it undergoes mitosis, it's spread out really thin because it's making these adhesive structures to the underlying extracellular matrix. However, if you want to divide, and you want to divide evenly, you have to round up into a sphere. And in doing so, then you can have a pole and you can pull this thing into two halves. So just at the moment it's ready to undergo mitosis, it lets go of 99% of these structures via modulation of the Talin linkages. And then the second it's divided, it immediately re-spreads back down onto its original footprint. So this is a coupling of two primordial systems, the cell division machinery and the cell adhesion machinery. And we're very interested to understand this. And we're also very interested to understand these adhesions. These are this is a synapse in our brain, how one neuron talks to another neuron. And this is scaffolded via a complex meshwork of these proteins, of which integrins and talin are essential components. If you knock out the ECM, the integrin or the talin, then you don't have synapses. So we're very interested in understanding all of these different structures which are mediated by talin. As this is a professorial inaugural talk, I wanted to just take a step back to how we got to working on this and how we got this deep understanding of what I'm hopefully convinced you is a deep understanding of these systems. And Tobias gave quite a spoiler for a lot of this, but it started from a degree in biochemistry, and then in 1998 at Sheffield, before moving to do a PhD at Leicester on drug discovery and the use of NMR to look for small molecule binding to proteins. Unfortunately and tragically, my supervisor was killed in a road traffic accident in 2000, and at the exact same time my secondary supervisor took a chair in Manchester. So in 2000 I got relocated to Manchester, to UMIST, where Tobias was, down the corridor there. We never spoke. I didn't know he was there until we came here. But it's a small world. And then after that, after I got my PhD, I stayed for a couple of years on a postdoc, which didn't really go anywhere. In fact, that whole time up north wasn't very successful from a science perspective. So in a jokey way, we thought that we flew north to breed. And that breeding program was super successful because we've got two amazing kids now who very proud of. But in 2005, I actually left academia and I went to work at AstraZeneca as a senior physical scientist. When I was at AstraZeneca, I was using NMR to screen for drugs, small molecules binding to proteins. But I also got set what sounded like quite a boring job at the time, but turned out to be a major change in my life, is that I was asked to test all of the available NMR software and work out which one that AstraZeneca should adopt across their whole business. And this led to a really lucky occurrence, because one of the software which was just up and coming was a NMR called CCPN analysis, of which Gary is now part of the development of version 3 of this. But back in the day, this was predominantly script-based. This was really complicated. But Dr. Igor Barsukov at Leicester had written some pretty cool scripts which semi-automated a lot of that process. So I got talking to Igor, and he sent me the scripts, and the data he sent me to use as the model data was an unprocessed set he'd just collected on talin. It was actually on this region of talin here. So for the last 6 months when I was at AstraZeneca, I was working on this protein talin, and it went really, really well, to the point that Igor said, oh, you should come down to Leicester and work with Dave Critchley on talin. So in 2006, we packed up again, and we moved down to work on talin with Dave Critchley, who's the world leader in talin biochemistry, and formed part of what was a really amazing team. It's what you aim for in your own sort of group. I've tried to name it here. So this is Igor, this is Dave, this is Alex Gingras, and this is Neil Bate. And those three were postdocs, and our mission was to try and work out what talin looked like and what it was doing. And this went really well. That was a much more productive time, having to get no papers from two kids up in Manchester. Here we've got no more kids, but we got over 30 papers in that time in Dave's lab. And we went from this understanding of what talin looked like with this sort of low resolution detail. After over that 6 or 7 years, we managed to work out the exact domain structure of this, and then using a combination of NMR and crystallography, we could work out exactly what this molecule looked like. And what we found is is that this head region, this firm domain, binds to the integrin, and then at the very C-terminus, there's an actin binding site which couples to the actin cytoskeleton. But between those two attachment sites, we've got these 13 switch domains, these 13 rod domains, which are all arranged in series, and all will experience force in this mechanical linkage. And in March, it was 10 years since we published this paper, and we solved the last 5 of these structures, and we reported and we renamed them R1 to R13. Anyone who looks at any talin literature prior to this paper, the naming structure was a hot mess. It was all over, but we realised there's 13, we could actually give them meaningful names. But something which we determined, and what that paper indicated in the significance statement, was that we found that Talin changes partners in response to force-induced conformational change. So that is complex to show. So one of the things we've done in the last year is we've made videos to show this. So in white I've got the Talin rod domain, and in yellow I've got a protein which binds to one of these domains when it's in this closed folded state. What was already known is that inside those folded domains were these red helices which are binding sites for the protein vinculin. So that was established. Vinculin comes when we stretch this, but once we discovered that on the outside of these domains there's another set of molecules which are bound, then we could immediately see how you could convert force into biological signals. You pull on this molecule via the actin binding or via extracellularly, and you drive a change in the conformation of this domain, and then that changes the signalling at that site. What I haven't got time to talk about today, which is shown here, is that this is vinculin flying in, and just the mere process of stretching Talin to expose those sites is sufficient to activate vinculin from a tight autoinhibited state. It springs open and binds. So once you pull on this molecule, you trigger a whole cascade of events which lead to global reorganisation of the cell. And just as an acknowledgement as I go along, this is over the last year with Sam Barnett, we started to try and make videos to visualise what we're talking about here, and these are on my YouTube channel. Sadly for me, but very nice for Sam, is Sam got poached by industry to work on the Million Genome Project, earning double what I do, and with a permanent contract, which was much better than his precarious postdoctoral life. So, but he left, but sadly, luckily we got a lot of these videos done, and we're finishing up a lot of his stories now. So as I was transitioning to independence, in 2013, we'd worked out the Talin structure, and we'd worked out this single switch. So my head of school said to me, what are you going to work on next now that Talin is done? And this was a view shared by pretty much everyone in the lab, because in 2013, Dave retired, happy that he'd sorted this all out, and all the others went to work on completely different projects. However, there's some things about Talin which made me think that there might be more to this than what we'd discovered so far. Not least because Talin is pretty much perfectly conserved in all animals. This is the amino acid structure of Talin. But as we worked out functions of Talin and identified the key residues which were doing the processes which this molecule was doing, what we could find is that these are all perfectly conserved between slime mould and us. Like every mutant which we've made, not every mutant, nearly all the mutants which we've made are also conserved in slime moulds. And it goes much further than that, because the domain structure and the order of those rod domains is identical in every single animal which has been sequenced to date. Not just these guys which are in the zoo, but going much further back. Since Talin appeared, it's always had this same switch pattern. And that, to me, with this high conservation, suggested this isn't just connecting integrins to actin. This is doing something much more profound. And that's what I thought. I thought there must be some sort of order to why these are in the same order, because if they were just linkers over evolutionary time, they get cut and shut and altered and changed, whereas this hasn't happened here. So in 2013, when I was at this crossroads in my life after finishing this, I decided that I wanted to understand this and understand why this Talin was so well conserved. So to do that, I initiated two experiments, which led to me forming two key collaborations with Guillaume Giacomet, who was at Manchester at the time, but now is got his own lab in Finland, to find what else Talin's binding to actually do proteomics on this and see what else is bound. And also to G, I don't think he would be too thrilled with that photo, but this is G, this is my friend in Singapore, and he just is an expert in single molecule manipulation of proteins. What's interesting is I was strongly advised against doing both of these experiments because proteomics was apparently being done by a group in San Diego, which never saw the light of day, and also that it would be impossible to collaborate with the Singaporeans because they wouldn't want to engage. Both of those, that was terrible advice, and I'm glad I didn't listen to them, because these are two of my best friends now. But the question was, we can see this guy binding here, what else is on this molecule, and also how hard do we need to pull on this to open that switch and trigger this cascade. And this is from a few years ago now. This is the experiment which we first did when I emailed G back in 2013 and said we'd discovered this switch. Wanted to know how hard we have to pull on this. So what we did was we take a single molecule of Talin, just one molecule, which we can attach to the cover slip of a microscope via its N terminus, and then by its C terminus we can attach it to a magnetic bead. And in doing so we can use a permanent magnet to pull on that bead and watch exert physiological forces onto this molecule and see how it behaves. And when we did that, this is the data we got, which is quite, I think it's amazing. Like, this is us pulling the molecule taut, so we're pulling it like this, so it's getting pulled longer. But as the forces increase, you see these jumps in extension. That's each of these three domains overcoming a force threshold and unfolding. So that means that each of these three domains is behaving independently. And this is six cycles done in turn. So you can pull this to a complete spaghetti, and the second you let go, it immediately resets itself back to that original switch pattern. And what I'm not going to show you today is using structural biology and looking at structures, you can identify which of these domains is which, and we can make mutations to change the order at which these unfold. But I'd just say this wasn't deemed referable, but this paper, because it's only in scientific reports, but this has been really influential because this has been cited over 350 times and showed for the first time this switch-like behaviour of these domains. So on the back of that, when that paper was under review, I came in this is cringy to look back at actually, but this was my job talk on the 23rd of February 2014, and we just found this switch and we were like trumpeting it, and this was the paper under review. And in this cheesy bit at the end, the long-term vision, I said it was to try and establish a major international research group at the University of Kent for the understanding of cellular adhesion. So I'm actually quite chuffed because I believe that I have actually done that. And also we said that the way we were going to get at this was to write a Human Frontiers grant to look at this. So this worked because on the 1st of August I started here at Kent and set up my own lab to look at Talin signalling. Sadly this didn't work. This got triaged at 30%, so we didn't get that one. But we repackaged it and reworked on it, and then the next year we brought in Anna Akmanova from Utrecht to build this team and make it stronger and to look at the link between adhesions and microtubules, which led to us getting this grant. And these people, I can't thank enough. These are great friends, and this collaboration's been absolutely amazing. This is G, slightly more flattering photo, Anna and Guy in Vancouver. And the team which we've assembled, it's just been absolutely amazing. This is a few years ago. This is Ming Chi, who did all of those single molecule stretching, who's just now set up his own lab in Sustek in China. So now that the travel bans to China are lifted, I'm hoping to go and visit him soon. But with Miao and Shim In, we've took this whole machinery apart and really built a detailed understanding. And what I'd say is that these two things need to go hand in hand. You can collaborate in a transactional style for one specific thing, but to have collaborations on the long term, you need to be friends with these people. So these are some of my best friends on this screen now, and they're fantastic collaborators. And what I'd say is I think that together we've changed this field. And with Guillaume's experiments, with what we were doing, we now had over 40 different ligands which are binding onto Talin, onto these domains. And when we repeat those single molecule experiments, we see this. This is a single Talin molecule which we're stretching, and we're watching how it responds as a function of mechanical force. And this is like 7 or 8 years old now, but it's still jaw-dropping to me. This is the behaviour of a single Talin rod region. You still see R3 unfold at the start, but then you see this step-like behaviour, and each of these steps is one of these rod domains acting as a binary switch. It's going from a closed state to an open state. And each of these steps, because these domains are binding to different things in the folded and unfolded states, each step represents a change in the shape of this molecule and a change in the mechanosensing, converting those forces into biological signals. And something else which we discovered in that paper, which I'm going to talk about at the end, is we discovered that Talin's a really effective cellular shock absorber. The reason being that you've got all of these switches in series, if you pull on it hard, then a lot of these domains can unfold and you get a lot of slack in the system. So for force to rebuild on this, it's then got to pick up all that slack. So this prevents the cells getting ripped off in response to force, and also that this can also change the signalling at that site. So this was the first time we showed that Talin was a cellular shock absorber. But back to what I want to talk about here, one of the things I've been really trying to do over the last few years is to develop a theoretical framework to understand mechanosignaling and mechanobiology, and this concept of mechanical signaling hubs. And this is shown schematically here, and this has been a lot of help from Martin Schwartz at Yale, G in Singapore, and Nick Brown in Cambridge, helping me to develop this theory and push back against me going too far into, ground it, which has been very, very helpful. And what it looks to is you've got these 13 domains which can be in this closed O state or this open 1 state. So depending on whether they're closed or open, you recruit different molecules onto that site. So here we've got 4 domains I'm showing just for aesthetics, and all of them are folded, and the readout is the different molecules which are decorated in that state. When we pull on this molecule and we cause the weakest domain to open, we change the biological signalling. The readout changes as a function of the switch pattern, because we displace one set of molecules and we recruit other molecules. And if you pull again, the next weakest domain opens and you change this signalling further. So this led to, in December of 2021, putting together one of these. These are my favourite type of review articles. These are the at a glance in Journal of Cell Science, where you make a poster which summarises the whole of your idea and in a way that is hopefully accessible. If anyone wants one, I've got loads of these on my desk upstairs. But if we look at the switches, then this molecule is switching on and off lots of different things, because all of these switches bind to different ligands. The only one we haven't got something for is R5, but this is the most conserved different. It's the most interesting bit. We don't know what it does yet in the whole molecule. But what this leads to is this idea that you could switch on and off different biological programs by these mechanical signals, altering these switches and switching on and off different programs. But what it also leads to is that this is getting really, really complicated, because it's not just a molecule which is attaching an integrin to an actin. You've got a network of switches which are all with competitive interactions, because there's multiple ligands for a lot of these, and you've got different ways that this is connecting to the cytoskeleton, and you've got the changing in the extracellular environment, the stiffness, the composition, all of this. And yet somehow, from the fertilization of the first egg all the way through, this works out, leading to this notion that there has to be some form of code underpinning all this, because this is incredibly robust, and it's also very metastable with all stochasticity and random events. This all still works out. So we're really interested to understand this code. And what I've talked about so far is purely on a single molecule of Talin. In a cell, that's not what it is. We've got many of these molecules which form a meshwork on the bottom of the cell. And it's not just Talin with switches, vinculin's also got switches. So you've got this complex array of mechanical switches on a cell. So I changed it from Talin code to a meshwork code, or what's now been shortened to a MeshCODE. But then depending on the environment, depending on the conditions, you've got a series of switches here with binding sites available for all of these different proteins, and they all get recruited onto this in a dose-dependent manner, as a context-dependent manner, to change the signalling of that cell. And this is what we're trying to understand. But all of what we've thought about in terms of cells comes from cells in culture. A cell in culture becomes migratory. It also becomes very dynamic. The cytoskeleton in a cell in culture is incredibly dynamic. And what that means is that you've got, when you consider these switch patterns, you've kind of got a mixture of these, because you've got changing extracellular environment, because this cell's moving across an unknown surface. And you've also got the fourth generation machinery from inside the cell pulling on this. So this means that you get robust patterns, but these are very variable and dynamic, because there's two contributing changing variables. So the big idea and the change in my thinking on this sat at Sydney Airport in 2019, chewing a croissant, thinking about a question from Peter Gunning. And he's a very insightful guy. We collaborate with him on a few things, and he likes to come and watch the cricket. But he asked a question that how can this mechanical code work? Because you talk about a single Talin molecule, but there's lots of them. How are they all working together? And that wasn't like a breakthrough moment, but it was trying to work out what's actually going on here. And it led to a pretty straightforward change in thinking, is that the system has to be ordered. All of the cytoskeletal connections, all of these links to the nucleus, there has to be some form of ordering in that, which goes beyond the current view of this very dynamic system. And in that case, you can ask a really simple question to frame that slightly differently. And that's that if the extracellular environment isn't changing, if the cell is where it's supposed to be and it's in its position in the brain or in the heart or in the tissue, then the extracellular environment could be much more constant and much more predictable. And in that case, as the reviewers hated me saying this isn't say it doesn't change. This is just saying that the cell's rebuilding this exactly how it wants it. It knows how it responds because it's built it with the help of the fibroblast. But in this scenario, now you haven't got a tug of war anymore. What you've got is you've got a series of switches which the cell is in full control on. And it can have a signal enter the cell and switch on its contractile machinery, pull on this and update this pattern. So that led to me getting home really excited, and I wrote a paper called Ordered Systems, and it was like really, really long and quite complicated. But on page 19, at the very end, I had a statement saying that it could be an exciting prospect to imagine this data storage being put to use in the brain. And that was just a throwaway comment. And Kate, who sat at the front, my wife, she said, she read it, she's been very helpful for reading all this stuff, and she said, are you saying you think that memory could be stored in the shape of Talin molecules? And then asked, do you not think that's quite important, that you've got a throwaway paragraph at the end? So that went straight in the bin. And then I went away and we rewrote it. And over the next year, with a lot of help from all of these guys, like Neil and Mike and Martin and many others, started to buff this into shape to try and get something which made a bit more sense. And in 2021, after over 7 months in review here, after being in review in other journals, finally this paper came out, and this is probably my best paper to date, although it still not fully took hold. But this is a mechanical basis of memory. This is to ask that idea, if these cells are in ordered environments, then the switch patterns, that binary string of information, you could use it to actually store information in these adhesion complexes, because you could write into these complexes in a binary format using small changes in contractility, as is shown in this video, and then that would change the signalling. So in that paper, it starts off just discussing this as a framework, and then it speculates about what this might actually mean. And that led to this idea that the brain could be a mechanical computer. Those scaffolding, those adhesions which are scaffolding all of these synapses, and which are essential to hold those synapses together, these switches are built into those synapses. So what would happen if you could write information into those switches in the synapses, holding them together? And then you could imagine that the signals which go across, and which loads of groups have shown trigger contractility, they switch on the motor proteins within for the pre and the post-synaptic side, that could be pulling and changing these switch patterns, but in also writing information, but also changing the activity of the synapse. That was the idea, which you could take or leave. But what we've been trying to do over the last couple of years is to try and prove if we could actually prove this and prove that there's a binary code in animals. And what we've been doing, and again this is with Sam Barnett, we've been trying to model this. We tried to build a simulation, but then we realised that that's really, really difficult, and we weren't equipped to do that. But when we were making the building blocks to feed into that simulation, we noticed something much more immediate and much more impactful, I think. And that's if you look at what these molecules look like when drawn to scale. This is a Talin molecule in a closed conformation, and when it's first stretched. When this molecule starts to unfold those switch domains, it starts to undergo these quantized step changes in length. And it goes, these are huge. This can be 800 nanometers and still bound to the integrin at one end and the actin at the other end. So you could have a mechanical linkage spanning almost a micron across a cell. And then if you look at those things binding to these switches, but draw them to scale, then you can immediately see what's going on here, not just recruiting the specific molecules to that site, you're also spatially positioning them in Z. So here I'm calling the membrane X and Y, and then Z the dimension going away from that. And here, like this is just showing 3 of the 13 switches folded, because otherwise they'll go down onto the ground floor. This is way down. But if you look at the different ligands, orange, green and pink, then in some of these are very close together, and in others they're spatially resolved, and in others they're freely diffusing. If the binding site's gone, then this green one's just diffusing around. But we're very interested now because we're very interested in the enzymes which bind to this, because those enzymes will be getting moved around within this compartment. And the best characterised one which we've done so far is CDK1. I don't want to go into too much detail on this, but just I wanted to show another nice video. But this is CDK1. It sits on this R8 switch of Talin, and it phosphorylates lots of different things in the adhesion complex. But one of the things it phosphorylates is ultimately Talin itself. When it phosphorylates Talin, it converts this R7-R8 switch from the strongest domain in Talin to one of the weakest domains in Talin, such that the next time you pull on it, if it's being phosphorylated, that will be the domain which will open first. And that unfolding will introduce 112 nanometers extension into this. So the geometry and the dimensions of this whole system will change as a function of these enzymes which are decorating it. But what that also leads to is, I'm trying to get more into this computational framework, is that if you've got an active kinase and you've got tension, then this will cause a 112 nanometer extension in this. If you've just got an active kinase, it will phosphorylate lots of things around it. And if you've just got tension, it will unfold the next weakest switch. So this is logic gate operations which are acting on a single molecule, in my opinion. And it gets to this framework where if these enzymes are held close together, it doesn't take much to imagine that if you bring an enzyme and substrate close together, it can activate that, it can have high activity. And similarly, if you hold them very far apart, then they can't see each other, they never come into contact. So you can have these same molecules within a very narrow, a very small area with very different activation just by positioning them differently. And this idea of signalling scaffolds isn't new. Anyone who's done a cancer undergrad module or done any secondary marking on one will have come across the kinase suppressor of RAS, which is in the MAP kinase pathway. And it's a long piece of spaghetti in the simplest sense with a few bits. But in essence, what I'm interested here is it's a long piece of spaghetti, and all of the MAP kinase cascade enzymes bind to it such that they can phosphorylate down with a really high activity. And this is a way to control the flow of cellular information. You bring everything together in space and time. What we're saying here is that Talin is mechanically operated. This is something which we're just trying to finalise now. But you're not just bringing them together in space and time, you're moving them relative to each other in space and time to control this activation. And if you look at what this looks like, I don't know why that doesn't play, it's a bit weird. So this is an example of something which we're working on in the lab. This is an enzyme which has got, I wonder if I can go back to the start. It's got an enzymatic domain, but it's tethered to the protein via a binding site here, and then a long piece of spaghetti. What that means is that that enzyme has got high activity anywhere within this sphere, much lower at the edge and in the middle, but I think we were talking to Neil about this, about a three-dimensional donut or something. But anyhow, anywhere within that sphere, this thing's going to be active, and it can phosphorylate its targets, or acetylate in this case. But what you can see in this arrangement is that the membrane-bound proteins won't be substrates for this in this arrangement because it can't physically reach them. And this is very much like this goat in a field. We used to have goats when I was a kid, and if you stake it in a field, you'll get a perfectly tight circular grass, which is the radius of the length of the rope around that stake point. If you make the rope longer, it'll lead to bigger circular grass, but if you move the tether, it will have access to a different patch of grass. Exactly the same is happening here. This enzyme, if we open another switch, it will have access to things over here. So you're spatially organising the enzymes in 3 dimensions, but predominantly in a single dimension. So we got quite excited by this as an idea, and we went away and we measured all of these zones of activity for known ligands for Talin, and measured how big they are on the scale drawings. Because what we want to do is we want to look how this would look inside the brain. And this is from Wikipedia, but this is a dendrite shown at the bottom. And it's got hundreds of these dendritic spines. These are the synapses. These are the most common form of synapses. And there's hundreds of them on each dendrite. But if you look at the dimensions, then they're about 1 micron by 1 micron in length. If you look at what Talin looks like drawn to scale on that, and we can use the vinculin binding site to define the maximum dimensions, then fully closed Talin is very, very small. Whereas when Talin's fully unpacked and engaged all of its vinculin binding sites, it can stretch to the full 800 nanometers and span this whole thing. But all of the stages in between, you've got different switch patterns with different spatial arrangements, because we can take those zones of activity, those goats, how big that goat could eat, but these enzymes, how big their sphere of activity is, and dock this onto this based on what we know about how Talin functions. And what you can immediately see is that with these switch patterns, different switch patterns organise these enzymes and substrates differently. And some of these, they're on top of each other, presumably with higher affinity, but then some of them, they're spatially segregated in this Z dimension. And it's overwhelmingly in the Z dimension because all of the talins are attached at this end and the contractility comes from the bottom. So this is spatially resolved in a single dimension. And this is precisely what we're now starting to be able to see. Looking at the length of talin and looking at the localization of molecules on talin, and this is work with Tony Koleski's lab in Yale. You can't see the guy's first name because that's on it. But Rob Nisha, we can measure this. Talin is much longer in spines than what it should be if it was fully closed. And it's also different in different lengths in different spines. So this is information, we think, written into these molecules. And what's also interesting here is that the green is always at the top and the pink is going away from it. So that really fits nicely. But what we start to imagine, I think this is near the end, is that we start to imagine that you've got a mechanically operated part of a machinery which can be moved up and down relative to a static part as a function of these binary strings of information. And it turns out that these spines are perfect for having a sealed mechanical unit. You can actually have mechanical signals going across a spine without any crosstalk from its neighbours because it's fully sealed off. And that's why I think they have this structure. But you could imagine that you could organise these molecules in these spines in a certain way, and that some of these codes would give high activity and some of them would give reduced activity. And we thought it was a bit like this gearbox model, because here this would be fixed until you give it a signal to change it. When you give a signal in the car, you change gear, you move the mechanically operated part relative to the static part and change the transmission across that synapse. So the idea which we're trying to prove with Tony and others is that this is what's going on here, that each of these spines looks exactly the same with a microscope, but they've all got highly different activation states because their enzymes are arranged differently as a function of these switch patterns. And if you could get on board with that, that the activity of each synapse could be controlled by these switch patterns, and that the signals, the electrochemical signaling, could change those switch patterns, then if you have lots of these spines, then you could start to write information in a useful manner. And then you could envisage that if you had an array of these mesh codes, then you could write data. So this is what I really want to try and prove. This is the big aim of our research. Is that if you look at what the cortex of the brain looks like, it's comprised of millions of these cortical columns. This is a beautiful drawing, by the way. I really admire this one. But each of these teardrops, they're incredibly repetitive, it's incredibly logical, and each of these teardrops has got 60,000 synapses. So this is a really complex wiring up of synapses and electrical wiring in the simplest sense to connect all of these together. If you look at what this looks like in terms of, I was struck by the similarities between what we've got here and the architecture which we've converged on as we've tried to optimize data storage in silico. In silico, we have the terabyte disk which fit on the end of your fingernail. There, you've got, it's made because you have many, many channels, all identical, but with different numbers, and then you physically write information to whichever one you want by directing the flow of electricity via a memory controller. I think that's exactly the same as what's happening here. The hippocampus or somewhere else maybe writes and reads information from this and stores all the information in the shape of these molecules is the big idea. But then if you could get on board with that, then here's a guy who's about to go for a shower. He puts his hand in to see whether it's freezing cold or boiling hot. But his sensory neurons send information into him via these action potential spike trains, little pulses of electricity down these wires, and the frequency of those is what sends information. But in this model, these electrochemical signals would be changing these switch patterns, which would be changing the direction and the flow of electricity around this complex computational device. And if you could imagine that, then if it's not just a guy going for a shower, every single thing you do, every single thing you experience could be written by sending these electrical pulses down the right wires to update these switches. And you could then start to predict that there's a mechanical representation of each animal's unique life could be written into this coding, which specialises it for its own unique environment. So just to ground it back, because I want to have a couple of minutes to talk about something completely different, which is equally wacky but in a different direction. This is based on experimental fact. We can prove that these are memory molecules. We can write information into them and store it persistently in the shape of the Talin molecule, and we can do that using small changes of force. We can show that they coordinate these enzymes binding to it, and so this is experimental at the single molecule level and into the cell level. What I'd say here in the brain is I'm proposing that information, the engrams, everything we learn, is written into the shape of these molecules in these scaffolds, changing the weights of each scaffold, of each synapse within a network. But the cool thing is that this would fit with the existing models beautifully, because the electrochemical signalling is darting around, everyone knows that. But the route it takes is a mystery. Here it'd be changing its route because it'd be changing the weights and the activity of each of these synapses. So I think that this is what we're hopefully going to prove over the next 5 years at Kent, is that this is an information processing system which we call our brain. But what I want to just pivot on is to talk about something which came from understanding these mechanical molecules and how they change how we are led to a step change in material science, I think, and so does many other people. And that's because this is best shown here. This is a cell migrating along the surface. You've seen this a few times. We've defined the machinery which this cell's using, and it's using this unfolding and refolding to coordinate the signals, and to also to buffer against forces. We could pull it so it gets five times longer. The tension stays low because of this stochastic unfolding and refolding. So here, this is defined this as a cellular shock absorber. And what we were very keen to do was to see, could we actually harness this shock absorbing property? These protein polymers behave different to human-made polymers. So could we use that in a way that we could make these new type of materials? And this is a collaboration with Jen. I don't know if she's here, but this is this started off as a wacky idea, and then it developed into a serious idea. And this was one of the very last GTAs that Kent awarded, was between me and Jen. And we were lucky to get an exceptional PhD student, Jack, who did most of the work on these next few slides. To make this material, go from this wacky idea and to actually design something which can capture these projectiles which have been fired at 3,000 miles per hour. And in some respects, the experiment is quite straightforward, and the chemistry is very straightforward. We take a fragment of Talin, we took this region because that's the region we've characterised to death. We engineer it so that we can polymerize it, so we functionalize this molecule. So we're going from a protein which is making E. coli to a protein which we're treating purely as a polymer. And then we can use cross-linkers which have got 3 heads to actually join this. So each Talin connects to 4 other Talins which connect to 4 other Talins to make what's called a hydrogel. And that's shown here on the end of this spatula. But instead of like normal hydrogels are built of polymeric proteins or different monomers, this one's got the intrinsic shock absorbing property of the Talin within its molecule, within its monomeric unit, which can undergo this large change in extension reversibly as a function of force. And if you look at what these look like using scanning electron microscopy, they've got this fibrous nature. They're incredibly sticky and they're very stretchy and they're transparent. And apparently transparency and stickiness are also very desirable properties in this sector which we're trying to move into. But what's interesting is that this polymer, this hydrogel, behaves like no other hydrogel which I've come across or which we've come across, in that it gets better each time it gets stressed, and also it does this behaviour, where normally with a hydrogel you pull on it until you break it and it starts behaving like a gel, and then it suddenly behaves like a solution. Here we see the gel, and then we see it go to what looks to be a solution until a certain point, and then it picks up the slack and starts to behave like a gel again. So this gel cell gel is, so far it's unique to these materials. And the idea there is that these switches, when you manipulate or apply strain onto a hydrogel or onto any material, you reorganize the fibre network. And in this case, what that leads to is more of these domains organised in series and in parallel, such that when it starts to experience force, it's stronger, but once it gets past the tipping point, there's a huge mass unfolding event. All of these domains open simultaneously. And then once the event's gone, it refolds. So that's why we get that mountain-like view of the gel to solid to gel transition, or our current idea. What I was very keen to show is, and this bothered the chemist, but I really want to show that this property of the gel was tail independent, because it might just be that we've made a gel which is cool, but doesn't. And the way we got that is to use a constitutively active vinculin, which is green, and this is some nice imaging from Karen Baker, to show that once you stretch tailin in the presence of vinculin, it binds and never lets go. So all the fibres get turned green, and they also lose this reversible refolding. And you can see that, because if you use just GFP alone, you can see the inverse. This is a tail-independent phenomenon which we're seeing within this material. And then a big shout out to Luke, who sat at the back, because then what we've got downstairs, this has been a nice collaboration with chemistry and with astrophysics, is that we've got the light gas gun which can fire projectiles at thousands of miles per hour. We chose a modest 3,000 miles per hour to do these experiments. And the experiment is you basically got a high-speed gun, and you have a cartridge which you accelerate, which hits this sabot full of beads, basalt particles, which get accelerated down and into the material sat in the target here. When we did this with the control sample, which we do our rheology on, it's completely obliterated, and what we see is that it sheds straight through the sticky tape and dints into the metal plate. When we repeat the same experiment with the TSAM, we still get a bit of damage, but by and large the backplate is totally unscratched. And the exciting thing for the astrophysicist, and for us I suppose, is that these particles were captured intact, sat inside this material. Normally once they hit, they're completely blown to smithereens, but here we could capture and retain them and actually analyse them after they've been hit, which has quite good implications for the capture of high-speed stuff. But that shred where we've got that tear is actually caused by this guy. This is a piece of aluminium out of the burst disc here, which has gone ricocheted in and embedded into our material. So we caught the shrapnel as well, which was quite cool. What was interesting is you put this out as a preprint and normally get a few retweets from some of the people in this room. But this one got incredible traction because it is a really cool idea. And it started to make it into the mainstream news cycle, and it's got a lot of interest. And the best interest we got was that the editor at Nature Nanotech emailed us that same day and said, "This is amazing, would you send it to us?" And, that's not come out very good. But this has been recently published in Nature Nanotech. It's going to come out next month. And in December 2022, at the end of that project, when we submitted it, Jack graduated. These two looking very happy, me looking relatively happy. But this was a really nice day. This was Brill. This was we submitted the paper, filed patent at the same time. But since then, we got funding from the Ministry of Defence via their DSTL, Defence and Security Accelerator, and we've been able to really grow this team. And we've got a fantastic team now, including Jen, Amber, Dr. Amber Paswani, Yuzumi, Luke, who, if you look at him at the back and look at him on this photo, he wins the Catfish of the Year photo award, even beating Neil at the front, and Perry. But we really took this and moving it on to the next step, trying to translate this into usable materials. So just to wrap up, just where next? I'd say that the next 10 years of the Galt Lab research, and the RPD and the appraisal people aren't going to like this, but it's going to be tailing, and more tailing, because there's so much to understand. The diversification of the portfolio is going to be from what tailing does in different conditions. And it's going to be in three main areas. I think we're getting incredible traction improving tailing's role in many different diseases, including cancer, aneurysms, heart disease, and capillary leak syndrome. And we've got good funding now from CRUK and British Heart Foundation to get at this. But we've also got new links to childhood epilepsy and Alzheimer's disease. So we're trying to understand these with a view to proving this. If we can understand epilepsy as a misregulation of this system, then that would maybe lead us in the direction of proving this. And then we're also trying to develop the materials and see how far we can push this. And it'd be very nice to spin this out into a company, perhaps, if we can get through some of the bottlenecks. So So I want to finish, and I've got 2 thank you slides. One is to thank, it's been a wonderful time at Kent in this last 9 years, and I've been very fortunate to have a very excellent people working in the lab. And with a few blips, this has been an incredibly wonderful and prosperous and productive time, and most of these have gone on to do really cool stuff afterwards. Special thank you to the lab who are all sat over there, nicely arranged in order, pretty much. But it's brilliant. The time in the lab now is absolutely fantastic, and I think we're pulling in a direction which is really exciting. I should thank Jen and her lab, because this has been fun, and the Jeeves and the Mulvihill lab, who we share a space, and we've got on really well in the whole time, which I think is really important. I think the
you.
Yeah, yeah. Apart from him. And he's a good neighbour as well. And I'd also like to thank the people who do it on the collaborative scale, because I think we've put Kent at the centre of a collaborative network working to understand tailing. And this is all the places where we've collaborated with all. And in particular, I'd like to thank the labs which we've currently got collaborations where we've got papers which we're currently writing, because it's truly a diverse, international, interdisciplinary way that we're going to answer this and solve exactly what these molecules are doing, and to understand what's going on here. So I'd like to thank all of these guys as well, too many to mention. And lastly, I'd like to thank you guys, because I think I've gone on too long, as always. But thank you for listening, and thank you for being great colleagues over this last 9 years.
Okay, any questions for Ben? We have time for a few. Thank you for a great talk.
Thank you.
Gary, you.
When you have molecular reactions, local concentration can be a huge effect. Could you actually imagine controlling the rates of reactions just by having enzymes on a vinculated tailin and unfolding and folding it?
Yeah, 100%. That's exactly what we're trying to show. That's a very good summary of what we're trying to show. Yeah, because that's what I think, you're moving these relative to their substrates and relative to their inhibitors as well, so you can coordinate.
Yeah,
you can have everything within that. One more time?
Do you think you could use that industrially as well?
Yeah, so there's really nice bio batteries I didn't know about till the conference was at last week, where they position all the enzymes on a mesh work. But here you could actually change the activity of those. So yeah. But there's a few bottlenecks on the translational path here still to work through before I start to do another one. But yeah, it's a good idea.
There was, I think you were next.
Has anyone looked for any domains on Talin that might be sensitive to A beta or alpha synuclein aggregation?
I don't know if they have that, because tailin's on the inside, AP. But yeah, it's a very good question, and we're very interested because tailin directly binds to APP. So we're interested in that as a notion of if mechanical signaling is getting corrupted via changes in APP processing. So yes, we are looking at that as a short answer.
Okay, we'll take one more question here from Jan.
Okay, let's give you a little challenge, Ben.
Okay.
So I love your theory about the binary code and the fact that this molecule might be able to store information, and you've done some lovely work writing these theoretical papers about how that is, but at the moment it is still a theory. So how are you going to go about testing it in vivo to see whether this is actually going to work?
Yeah, so that's exactly what we're doing now. We can see these shape changes in neurons in culture. We can actually stimulate neurons and do limited proteolysis and see that tailin has changed shape dramatically across in the untreated and the treated cells. And because we can physically measure this, these lengths in cells, but it might be nice to actually look in worms, I think. Is that what you... Because there you've got the whole connectome, so if these molecules are changing and the information's going around, and that's one of the things we're doing with the guys in Bordeaux, actually make readers for this, to read the shape of these molecules fluorescently tagged, so then you can read what shape the molecule's in and see. So yeah, I think that's the next challenge, and that's why it needs to be collaborative, because we don't have a big neuroscience grouping here at the minute. But yeah, that's the goal for the next few years.
So I think we'll have to freeze the lecture theatre for the lecture, but there's coffee and noodles in the hall artist room, and you can continue your questioning there. also unusually organized since I was already an